Organoid intelligence is a relatively new field of research that is rapidly gaining momentum in the scientific community. At its core, organoid intelligence involves the use of three-dimensional cell cultures to study biological processes and gain insights into human disease. These organoids, which can mimic the structure and function of specific organs in the human body, offer a powerful tool for researchers to better understand the complexities of human biology. The concept of organoids dates back to the 1900s, but it wasn't until the early 2010s that researchers began to develop methods for creating organoids in the lab. Today, organoids can be created from a variety of tissues, including brain, liver, and kidney, among others. These organoids can then be studied to better understand how different types of cells interact with each other and how diseases develop. One of the key benefits of organoid intelligence is its potential for personalized medicine. By creating organoids from the cells of individual patients, researchers can develop more targeted and effective treatments for a variety of diseases and disorders. For example, a patient's cancer cells could be used to create an organoid, which could then be used to test potential treatments and identify the most effective course of action. Another potential application of organoid intelligence is in the development of new drugs. Traditional drug development typically involves testing potential treatments on animal models, which can be time-consuming and may not accurately reflect how the drug will behave in humans. By testing drugs on organoids created from human cells, researchers can gain a more accurate understanding of how the drug will behave in the human body, potentially speeding up the drug development process and reducing the need for animal testing. Despite its potential, there are still some challenges associated with the use of organoid intelligence. One of the primary challenges is the difficulty in creating complex organoids that accurately mimic the structure and function of real human organs. Additionally, organoids can be difficult to manipulate and study, making it challenging to develop standardized methods for creating and studying them. Despite these challenges, organoid intelligence is a rapidly growing field of research with the potential to revolutionize the way we study and treat diseases. As researchers continue to develop new methods for creating and studying organoids, we can expect to see more breakthroughs in this exciting field in the years to come. Organoid intelligence has already yielded some promising results in the study of human disease. For example, researchers have used brain organoids to study the development of Alzheimer's disease and Parkinson's disease, among others. By studying the progression of these diseases in organoids, researchers have gained new insights into how they develop and potential new treatment strategies. Organoid intelligence is also being used to study the effects of environmental toxins and pollutants on human health. For example, researchers have used liver organoids to study the impact of exposure to certain chemicals on liver function, potentially identifying new ways to mitigate the negative effects of environmental toxins. In addition to its potential applications in medicine and drug development, organoid intelligence is also being used to study fundamental biological processes. By creating organoids that mimic the structure and function of organs like the heart, researchers are gaining new insights into how the heart develops and how it responds to different stimuli. One of the most exciting aspects of organoid intelligence is its potential for collaboration across different fields of research. By bringing together experts in biology, medicine, and computer science, organoid intelligence has the potential to revolutionize the way we study and treat disease. How organoid intelligence is different from AI. Organoid intelligence and artificial intelligence, AI, are different in several ways. Origin: organoid intelligence is based on biological systems, specifically three-dimensional cell cultures that mimic the structure and function of organs in the human body. AI, on the other hand, is a computer-based system designed to mimic human intelligence. Purpose: organoid intelligence is primarily used to gain insights into biological processes and study human disease. AI, on the other hand, has a wide range of applications, including machine learning, natural language processing, and robotics. Data: organoid intelligence relies on data from biological systems, while AI relies on data from a wide range of sources, including sensors, social media, and other digital platforms. Learning: organoid intelligence involves observing and learning from biological processes, while AI involves training algorithms to learn from large datasets. Complexity: organoid intelligence is focused on the study of complex biological systems, while AI can be used to analyze and model complex systems across a wide range of disciplines. While there are some similarities between organoid intelligence and AI, such as the use of data and machine learning algorithms, the two are fundamentally different in terms of their origins, purpose, and focus. In conclusion, organoid intelligence is a rapidly growing field of research with the potential to transform our understanding of human biology and disease. By creating organoids that mimic the structure and function of human organs, researchers can gain new insights into disease processes and develop more targeted and effective treatments. While there are still challenges associated with the use of organoid intelligence, ongoing research is likely to yield more breakthroughs in this exciting field in the years to come. Some frequently asked questions, FAQs, about organoid intelligence. What are organoids? Organoids are three-dimensional cell cultures that can mimic the structure and function of specific organs in the human body. What is organoid intelligence? Organoid intelligence involves using these three-dimensional cell cultures to gain insights into biological processes and study human disease. What are some potential applications of organoid intelligence? Organoid intelligence has potential applications in personalized medicine, drug development, and the study of fundamental biological processes, among others. How are organoids created? Organoids are created by using stem cells or other cells to grow three-dimensional structures that mimic the structure and function of specific organs. What are some challenges associated with organoid intelligence? Some challenges associated with organoid intelligence include the difficulty in creating complex organoids that accurately mimic real human organs, as well as the challenges in manipulating and studying these organoids. How is organoid intelligence being used to study disease? Organoid intelligence is being used to study the development and progression of diseases such as Alzheimer's disease and Parkinson's disease, among others, by creating organoids that mimic the structure and function of the affected organ. What is the potential for collaboration across different fields of research in organoid intelligence? Organoid intelligence has the potential to bring together experts in biology, medicine, and computer science to revolutionize the way we study and treat disease. How does organoid intelligence differ from traditional methods of studying disease? Organoid intelligence allows researchers to study disease processes in a more realistic and physiologically relevant environment than traditional methods such as cell cultures or animal models. What are some ethical considerations associated with the use of organoids in research? There are ethical considerations surrounding the use of human-derived cells and tissues in research, as well as concerns about the potential for organoids to develop consciousness or be used for purposes such as organ transplantation. How might organoid intelligence impact the future of medicine? Organoid intelligence has the potential to revolutionize the way we diagnose and treat disease, allowing for more targeted and personalized treatment strategies. It could also lead to the development of new drugs and therapies based on a more comprehensive understanding of the underlying biological processes. Are there limitations to organoid intelligence? Yes, there are several limitations to organoid intelligence. For example, it can be challenging to create organoids that accurately mimic the complexity of real human organs, and it can also be difficult to manipulate and study these structures. Additionally, there are limitations to the amount and quality of data that can be obtained from organoids. How might organoid intelligence impact drug development? Organoid intelligence has the potential to improve drug development by providing a more physiologically relevant platform for testing drugs. By testing drugs on organoids that mimic specific organs or disease processes, researchers can gain a better understanding of how the drugs will behave in humans and potentially identify new treatments for diseases. How might organoid intelligence impact our understanding of the brain? Organoid intelligence is already being used to study the development of the brain and diseases such as Alzheimer's disease and Parkinson's disease. As research in this field continues, it is likely to yield new insights into how the brain works and new treatment strategies for neurological diseases. What are some current limitations of organoid intelligence in the study of disease? Some current limitations of organoid intelligence in the study of disease include the difficulty in creating organoids that accurately mimic real human organs, the limited availability of human-derived cells and tissues, and the ethical concerns surrounding the use of these materials in research. What are some potential future developments in organoid intelligence? Future developments in organoid intelligence could include the creation of more complex and sophisticated organoids, the development of new technologies for studying and manipulating organoids, and the integration of artificial intelligence and machine learning to better analyze and interpret data from organoid experiments.